Good morning, students. I welcome you for the biology class. In the previous video, we have studied about cell wall, structure and function, cell membrane, structure, fluid mosaic model, and function of cell membrane. Today, let us discuss about the transport across plasma membrane, that is cell membrane. Substances may cross a membrane with or without the expenditure of energy. So there are two transport basically, passive transport, active transport. Passive transport, there is no requirement of energy, just the molecules move, whereas in active transport, energy is required where the molecules move from outside or from the inside of the cell. Coming to passive transport, diffusion, facilitated diffusion, osmosis. So they doesn't require energy, whereas active transport, pumps, sodium potassium pumps, as well as co-transport, they do require expenditure of energy. Understand, passive, it just move without the expenditure of energy, whereas active, there is an expenditure of energy. So here's an image for you. Diffusion, again from higher concentration to lower concentration. Whereas facilitated is also from higher concentration to lower concentration, but you can see a difference. There is a protein which is required. Some channel is required for them to move. Until that, the facilitated diffusion will never happen. Okay, some protein channels are required so that molecules can move. Whereas active transport, it is movement from lower concentration to higher concentration. So this is low and this is high concentration. So molecules are moving from low concentration to higher concentration with the expenditure of energy. And there is a requirement of some protein. Okay, protein channels like permeases is required, some pumps is required to transport. Active transport. So molecules move against a concentration gradient, that is from lower concentration to higher concentration. Cellular energy ATP is required. Example like sodium potassium pump and proton pump are the examples of active transport. So this proton pump and all you'll be studying in your higher class 10th in case of nervous system. Whereas passive transport, molecules move along the concentration gradient, that is from higher concentration to lower concentration. No cellular energy is required, whereas diffusion, osmosis, and facilitated comes under the passive transport. First, let us learn diffusion. It's a process of movement of substance from region of higher concentration to a region of lower concentration until a uniform concentration is achieved. Example, during respiration, due to the difference of concentration of carbon dioxide inside and outside the cell, carbon dioxide is given out as a waste product, moving from higher concentration to lower concentration. Similarly, where oxygen enters a cell by a process of diffusion, when level of concentration of oxygen inside the cell decreases. Coming to osmosis. Osmosis, it's a process of movement of water from its higher concentration to its lower concentration through a semi-permeable membrane. Osmosis is only a movement of water, that is solvent particles. So different types of solution exhibits osmosis. First one is hypotonic solution. First, let us know what is hypotonic solution. If the medium surrounding the cell, so when you keep a cell, the media which is surrounding the cell has a higher water concentration compared to cell. So inside the cell, water content will be less. Outside, the water content will be more. So this is known as hypotonic solution. So if the solution is very diluted solution, then the cell will start gaining energy, that is water. So they gain water by osmosis. Such diluted solution is called hypotonic solution. Okay. Endosmosis. Though water molecules are free to pass across the plasma membrane in both the sides, you can see this is getting in, this is getting out. There is a movement. So water molecules are getting in, water molecules are moving out. But you can see the net movement where the net movement of water is more inside. It is moving inside. Hence, well, cell will start swelling up. There is increase in the volume. This is known as endosmosis. Next, isotonic solution. If the medium surrounding the cell is of exactly the same concentration, that is water concentration as inside. So whatever water concentration is there inside, it will be matching to the solution. So there is no net movement. Okay, you can see in this picture two molecules are moving out and two molecules are getting in. So there is no net movement of water across the plasma membrane. So there is no change in the cell size. Such solution where there is exactly the same water concentration of the cell is known as isotonic solution. Iso, it is same. Next, hypertonic solution. If the medium surrounding the cell has lower water concentration than inside the cell. So outside the cell, you can see less water content, whereas inside the cell there is more water content. If the solution is highly concentrated, then the cell will start losing water. You can see there is only two inlet of water molecules. You can see there's four water molecules which is moving out, just to make you to understand. So the net movement is always moving out. Hence the concentrated solution is called hypertonic solution. And you can see the cell. The cell will start shrinking. Here's a picture for you. So it's an endosmosis hypotonic solution in case of animal cell. This is same. This will be normal. So there will be no change. Whereas in hypertonic solution, when you just keep the animal cell, we'll say it as shriveled or shrinked. In case of plant cell, we'll say as turgid. So when more and more water gets in, and this will be normal. And coming to this plasmolized, in the sense you have already learned about plasmolized in your previous video, you can say it starts shrinking. Next, osmosis with an egg. Remove the shell of an egg by dissolving it with dilute HCl. The shell is mainly made up of CaCO3. A thin outer skin now encloses the shell. So first part is that. So dissolve the egg shell while adding dilute HCl, and you have a just thin covering on the top, and this put this egg which has a thin skin and a pure water, and just leave it for 5 minutes. So what do you see? The egg swells because there is endosmosis process. Now, place a similar de-shelled egg in a concentrated salt solution or sugar solution. So, where water content is less. So, it will become a hypotonic solution. So, when you just keep those eggs in that and observe for 5 minutes, then you can see the egg starts shrinking because water passed out of the egg solution into the salt solution, the concentrated solution. So, it shrinks. So, here's an image for you. So hypotonic solution, when you just keep it in a hypotonic solution, the egg bulges. When you just keep it in a hypotonic solution, it shrinks. And there's a normal egg. You can also do this activity with the dried resins. So do it at your home. Now let us learn the difference between diffusion and osmosis. Diffusion takes place in all medium, means in any medium like gases, liquids and solids, whereas osmosis takes place only in liquid medium. Diffusion does not require semi-permeable membrane, whereas osmosis requires semi-permeable membrane. In case of diffusion, it is dependent on the free energy of the molecules. So it depends on free energy of the molecules which is diffusing. Whereas in osmosis, it depends on other solute particles which is also present in the system. And in case of diffusion, you can expect equilibrium can be attained. So until equilibrium is reached. Whereas in osmosis, equilibrium in the free energy of the solvent molecules is never achieved. So equilibrium cannot be achieved, or it is not a mandatory process to acquire an equilibrium method. Okay. Next, mediated transport. Plasma membrane renders the transport of many molecules of biological significance. Such essential molecules are moved across the plasma membrane by a special protein called transport protein or permeases. This process of forced diffusion of certain substances through the plasma membrane is called mediated transport. Some special proteins are required where the molecules move through this. So permeases used in the process are quite specific to the transport of a substances. Types of mediated, facilitated. So you saw in the diagram where a protein channels are there through which the molecules will diffuse. If not, the molecules cannot diffuse. Active transport also. There was a protein channel where it was moving against the concentration gradient. Coming to endocytosis. What is endocytosis? It's a process of ingestion of a material by the cell through the plasma membrane. There are three similar processes like phagocytosis, means cell eating, photocytosis or pinocytosis, and receptor mediated endocytosis. Coming to phagocytosis, it's a method of intake of food materials by certain organisms like amoeba. The flexibility of a cell membrane enables a cell to engulf the solid particles of food or other substances from its external environment. So here's a picture. This is phagocytosis where solid particles is engulfed, and that's how it is as a food vacuole which is formed. And pinocytosis, it's cell drinking, minute particles. Receptor mediated, we'll learn and still more in a higher classes where receptors will be there, particular substance will bind, and that's how a collagen protein vesicles will be formed and that will be taken inside. And coming to exocytosis, it's opposite to endocytosis. Endo, it is getting in. Exo, moving out. It's a process where membrane of the vesicles can fuse with plasma membrane and extrude its content to the surrounding media. This is also called cell vomiting. Okay. So, what are the things which is removed out from the cell, like undigested residues and the substances like hormones, enzymes, and there's a transport of substances completely across the cellular barrier, so that you can transport across the cell. Here's a difference of endocytosis and exocytosis. You can see the molecules getting in. So there is an invagination of the cell membrane and that's how the vesicles will be formed. Whereas in exocytosis, these vesicles start fusing with the plasma membrane and that's how the contents will be extruding out. Here is an assignment for you. Why does the skin of our hands shrink when we wash clothes for a longer time? Or will you just be for more than like 30 minutes or something in your washroom and just keep your hand in water and just observe. So what happens to your hand? So there'll be a wrinkling, there'll be a shrinking of your hands. Why is that so? Think, do the activity. Just take one mug or one tumbler of water and keep your hand for a few minutes and just see. So there should be a shrinking. So find out the reason why there's a shrinking of your hand. Thank you. Thank you. We'll study about the cell organelles, their structure, function in the next class. Thank you and have a nice day.